Hello everyone, welcome to this short quick module on computation in neural engineering. Here we'll see some of the basic aspects of mathematical analysis in neural science, and it will be a short quick module but very useful when it comes to your project implementation or any brain computer interface based or event related potential based project. So before showing you the analysis part, I will quickly provide you the recap. This is what we have already learned in one of the course, that will generate a stimuli followed by signal acquisition, which will be wirelessly transmitted to a computation system, and where you process and make the sense out of it, assess something, and it based on the application your final assessment varies. If it is like attention, cognitive decline, there can be this kind of 3 indicators, whether a person should consult a neurologist, whether it's inconclusive or repeat the test, or good cognition, right? Or if it is hearing screening, then a baby or a person can hear, person cannot hear, and there is some issue. Same way, a small aspect which we are not touched upon too much was telemedicine, that once this is diagnosed, you can transmit it securely to a clinician or a person who is placed at a remote area, mainly useful in resource constrained area or villages, something like that. So this was like overall approach. Today the entire emphasis would be on this third part, which is on signal processing. How we will achieve that, how we will make a neural sense out of the recorded data. So I will emphasize, as I mentioned, on this point number 3. What do we do in that particular point number 3? So in that 3, these are some of the basic building blocks which we will be implementing in a software called MATLAB. So when I say MATLAB, along with MATLAB there are some of the plugins which will be used. So that I will discuss it in the upcoming demonstration, a quick demonstration code walkthrough. Here you see this is like some of the basic steps written for signal processing aspect. What we will do is we will acquire biopotentials, response, and stimuli acquisition. Stimuli acquisition means all your triggers, at which particular point of time which kind of trigger was provided, right. So that is very important. Along with biopotential, you should also record a trigger. So that is for that particular thing. Also, once you are you have to apply filters to make a neural sense out of it. You have to remove the non-neural component, for which you can use 50 Hz notch filter. Now 50 Hz notch filter, why? Because in Indian setting electrical power supply is 230 volt 50 Hz. When you talk about Western world, when there is a 60 Hz power supply, you have to replace this notch filter with 60 Hz power supply. Prior to that, you need to make sure that you are not getting any kind of power or interference. You have to shield your wires which are guiding or taking your biopotential from your scalp to the acquisition system, should be completely prawn free. Also you can use a band pass filter, because most of the scalp recorded potentials which comes for our application or normal cognition application should be recorded within this particular point of range. When I say that, based on application, this range differs. If I am recording auditory brainstem response, this range would be different. So based on your target application, range of your filters changes. Also there is Eventlist creation. What is that? I will come to that and quickly walk through. You have to do some epoching, baseline correction, artifact rejection, and bin operation. Okay. So this is like an overall idea of what we are going to do or going to implement. Prior to moving to the code walkthrough, I will just quickly tell you what is the experiment. Experiment is on visual evoke potentials to assess working memory and all. So you can see here 3 type of images has been shown to a particular subject, right. So there are 3 type of images, which one of them is more frequent, which is the blue one, and other 2 are the rare or kind of less probable or less occurring images. So there are stimuli design is also another aspect which we can itself go into the detail, but for now, for the code walkthrough, what we have been doing, it is an experiment conducted to assess working memory and attention, which is being measured using P300 event related potential, which is happened when you have 3 types of images: standard, target, and distractor. Subject is asked to click on a mouse or count the number of occurrences when target images come. Let us say in this case particular red rectangle is the target image, then in between yellow image also comes. So then what is the significance for that? So that is your distractor. It tries to distract you, and you are not supposed to click, or you are not supposed to respond when the distractor comes. So it also checks your runtime memory and also attention. So this is very briefly the overall experimental protocol. How many electrodes have been used? You can see here 3 electrodes have been used, which generally placed on CZ, PZ and OZ, because this is a visual experiment. I have already discussed that for visual experiment your electrode should be placed in your backside, either central or occipital or parietal region. Okay. So today more focused is on code. So I will quickly switch to the code walkthrough. So this is what the recorded file looks like. Now I will just show you what is there. This was recorded using OpenBCI Cytone board, so which has a 250 hertz of sampling rate, which means that every 4 millisecond you will get one sample. So it is for one particular subject, subject 1, and it is opening. You can clearly see once it gets open that it will record 8 channels. Yeah, it is taking little, yeah, you can see this. So here you can see total 8 channels, 250 hertz. Also it is in comma separated format. These all are headers which you can see here. Sample index, external channels 1 to 8, 0 to 7 means 1 to 8, then accelerometer channels, other things are basically your digital triggers, okay, analog channels, timestamp and all this thing. Okay, this is mainly this is like your original timestamp. This is a timestamp formatted. So if you see the same thing is being followed, and this is some data here, some data here, some data here. Again, this all are like same thing which is not connected. So based on just a rough observation, visual observation, I can say that yeah, channel data might be there in this 3, 4 and 5 channel, because this is your first channel, second channel, 3, 4 and 5. So while acquiring data, we need to take this data, and then also you have to take digital triggers which will be a part of here. So I have already extracted this and data is saved here. Okay, so let us quickly load that. So you can see this data is stored here, which has some 2 lakh samples into 6. Now what are the 6 thing? First 3 are the biopotentials, and the other 3 is mainly used to give the triggers. Okay, before we start performing experiment, we need to make sure that EEGLAB is added to the path. Now when we upload this video, in description or in comment, I will put a link to my previous introduction to EEGLAB video, which will help you just to from where you can install EEGLAB. What is EEGLAB? It is developed by Swash Center for Computational Neuroscience to play with EEG signals. Okay, it can be of any system. Currently I mentioned OpenBCI Cytone system. It can be of any other system as well, using which you can take the data in EEGLAB with different plugins and play with it. So I have to add this particular EEGLAB into the path. So EEGLAB is added here, it is updated, latest 2022 version. You have to add to the path with subfolders. Once that is done, we can come back to the data. Yeah. So now what I will do is I will open the EEGLAB. I will show it one approach using GUI. I will also inform how you can perform batch file processing and all. In both cases you can do pre-processing, in both cases you can do feature extraction, in both cases you can do classification, in both cases you can do ERP extraction based pipeline, everything. Whatever you can do in GUI, same thing can be converted into a code, and then you can give a provision of batch file processing. Okay. Currently there are research going on online processing as well. So for that, online means current data is recorded and then we are processing it offline. How can we record the data and similarly process it at the same particular time? So that is research and which is currently going on to have on-chip filtering, on-chip pre-processing and on-chip signal processing. Okay, it also considers how easy you can have a how easy you can get computations and how you can decide a trade-off between hardware and software, because right, filters can be implemented using hardware, filter can be implemented using software also. We'll see how to implement using software. So in both cases you can apply filters. So how what would you prefer? in hardware filter also there are active filters and passive filters. Which one will you prefer? You should have answer for all these things before you perform any kind of hardware software code design, right? Software is very easy, you just take the data into system and perform it. So why shouldn't we go with software, or why shouldn't we go with hardware? Hardware will give you more precision or something. That depends. What is your roll off factor, what is your pass band age gain, what is your stop band age, so many things are there, right? So also your I have just talked so far about amplitude of the amplitude response of filter. Also you should consider is phase, whether your filter gives you linear phase or not. And filter is just one example. You are doing so many several things, and currently I am showing it for 3 channels. The existing microelectrode arrays uses more than 3000, around 3000 channel, 4000 channel. How will you take care about the computation of those 4000 channel? How will you make sure that out of this 4000 channel this much information is only useful, the other things that we don't have to consider? So all these things are happening, and currently in research, and which is used for online signal processing, on chip signal processing rather, where electrode would be on your head. Along with electrode there will be one small processor or signal processor, DSP what you say, it will be also placed there, and using that you can extract the signal. So coming back to this code walkthrough, here you can see I have already added EEGLAB into the path, and then we will run the EEGLAB. EEGLAB is a plugin to MATLAB, and as I mentioned, specifically designed for neural signal processing. So this I have installed 2022 version. If you have latest version, you can upload it. This is the home screen. Okay, it is like a normal software which has a horizontal menu bar with a title, nothing else. You can take the data from this file. Other options will get enabled once you have the data. So currently we have a data stored in MATLAB here. As you can see, it is a .mat file, which is a MATLAB array. So I have to acquire it using this file, import data. So using EEGLAB functions and plugins. First one is only MATLAB array or ASCII file. So I have to just select this MATLAB array. It will ask me few questions. This is pop import data. So when you convert this into a code, you have to write the line for pop import data. Okay, here it will ask me that from where do you want to get the data. So we have already this data ready. Okay, .mat file. Also we can give is sampling rate. So as I mentioned, it is recorded using Cytone. So it records at 250 hertz. If you have higher sampling rate data, you can go ahead with that. This also depends on your application. What is your application and all this thing. Okay, so I am giving this 250 hertz for now, and I am importing the data. Okay, so that no current data set will be converted to whatever data set I will give the name. I am giving the name of S1. Okay. So that is S1. Also you can get all the information. How many channels are there? 6 channel. What is the frames per epoch? Epochs, currently we have not entered any events, and also entire thing will be converted as one epoch. So that is one epoch. Currently we have not added events, so there are no events. This is 250 hertz. When it starts, when it stops. If you can see, total 842 second the experiment lasted, around 14 minutes. Okay, you can work on that. It has stored using 6.8 megabytes. Okay, so now one by one we'll see. As I mentioned, 6th, 5th and 4th are the channel which has the event. So we, after importing your particular data, we need to import event as well. Okay, so let's add event one by one from the data channel, because we have already given event information in terms of channel only. So we are adding it from data channel. Here you can use the 6th channel, because as I mentioned, 4th, 5th and 6th has event, 1, 2 and 3 has EEG data. Now what is trigger information or event information or event, and what is EEG data I already covered in the previous lecture. So yeah, this is like any previous event have been stored. Do you want to delete it? So when you are entering your first time event, you should delete it, and you don't want after that, once event has been extracted, you don't need that channel. So that's why this both the things are checked. Now all events are of same time, same type. For 6 we can say, but then some more events are also there. So you can keep that as it is. In this extensive help is available in the website. So whenever you have any question, anything, you can go back and check it that particular point of time. I am adding this particular event. So now this channel from frame will change to 5, because we have extracted the event information out of it, and now this is 5. Also you can see 100 events have been added. So that 6th channel had 100 events in between. Same thing we will do it for 5th channel as well. While entering the 5th channel, we need to make sure that we can't delete old channels, because we have already given information of 100 events prior. So that we should not change, that we should not check, but the delete event channels we can check it, because we don't want that channel to remain there. Okay, we only want biopotential when the channel is extracted. How we can make sure the channel is extracted, I will show it to you. So this is again 5th channel is extracted, this has 4 now, events has changed to 100 to 200. So both the channel has 100 occurrences there. Okay, further again for 4th channel we have to do the same thing. When you import the 4th channel, again you should not delete the old channels, say okay, then channel will be 3 and total number of events are 1000, which you can see here. So now this event has been extracted. Now if you see the data, how it is looking like, okay, ultimately it is an EEG data, right. So if you scroll through, you can see the channel, events are there, right, and 3 channels, but we are not able to see the EEG channels and all. That is because it is a raw data, it is not processed or filtered. You can say it is almost like a flat line. Now all this thing has a lot of EEG variation, how will we able to extract that, right. If you see, you are not able to extract anything out of this. All thing you can see is there are different event occurred, channel 4, channel 6, whatever was there, that has been extracted, you can see event, but how can we make a neural sense out of it, right. So there what you can do is, you have to see that what is the current frequency spectrum, that you can use plot amplitude spectrum from the ERPLAB function. Again ERPLAB is a plugin which should be added into the path in EEGLAB, which I have already added. How to install ERPLAB? As I mentioned, I will put a link of my previous video in the description or comment. You can go through that, because this is like an advanced course, so I am not going too much detail or I am not informing too much basic things, right. So this I will quickly plot the amplitude spectrum here, and how many channels you want to plot? First 3 channels. Now what is the upper frequency? 120 automatically. Why? Because your sampling rate is 250 based on Nyquist theorem. What is Nyquist theorem? Already covered. If you do not know, please go through and check it. What is Nyquist theorem, right? When your maximum occurring frequency is FM, your sampling frequency should be at least greater than 2 into FM. So let us plot the amplitude spectrum very quickly, and in amplitude spectrum you can see there is, this is one of the biggest bottleneck when if you are doing neural signal processing and all. I will show you using the data tip as well. 49.96, this is our main bottleneck, which is a power line artifact. How to remove that? We need to put a filter. Now 2 ways, one is put a notch filter and remove this, other one is put a filter from 3 to 30 or 0.5 to 30 and remove it. So we will currently put a filter from 0.5 to 30. How to put it? There is a function in MATLAB tools, filter the data in EEGLAB, basic filter, okay. So again I am here, there are you can feel free to put 1 to 30, 0.5 to 30, 3 to 30. For now I am putting 3 to 30, okay. Also for illustration purpose I am keeping this plot frequency on. So when I do that, you can see amplitude spectrum and phase spectrum also, how it performs the filtering. You can also see it is a linear phase during the range of acceptance. It is very important. If it is non-linear phase, there will be a multiple issues with delay, phase delay, all this thing. So it should be linear phase. Also you can see in this particular range your gain is 0. Now 0 means your signal will get 0? No, the dB is 0. When dB gets 0, log of something is 0, which is 1. So it will allow the signal with the gain 1, whereas other signals you can see is getting attenuated by at least minus 60 or minus 70 dB, which is of a huge contrast. So then it will allow the signal only within this particular range, okay. So I will have a very important naming convention. Once you filter it, you give the name, so you will have an idea that which data set, because ultimately all the data set will be saved here. So which data set you want to go back and redo some operation. First one is my original data set, then it is a filter data set, okay. So this is now again, let us see the amplitude spectrum, whether that small spike at power line amplitude has vanished or not. So again I am plotting it, you can see it is not there. So now whatever data is there, we believe can be of a neural value, okay. So next step is to create an event list as per the block diagram what you have seen before. Event list can be created using ERPLAB menu, create event list, and then you have to define your 3 events and bins. So I have already written it here. The first one is event code 1 for channel 4, which is my standard. Second one is event code 2, which is channel 5, which is my target, and the last one is 3 channel 6, which is my distractor, okay. Based on that I will be getting my values, I will be getting my response. So you have to just apply this particular thing, you have to take care how to write this event information, Once it is done, they'll ask, do you want to code the labels? So you can see, yeah, apply code the labels, and then automatically it will be renamed with dot dash hyphen or underscore e list prefix, and you will get one more data set here. So now you have events. So let's see how much change these two things have made. So earlier you are not able to see the data, right? Now you are able to see the data. You can play with this time range and all this thing. You see, keep it here 10. Okay, this might be due to some idling or some artifact that it will get rejected in artifact rejection upcoming step. So you can see based on the timing information and how your brain reacted to this. Here the scale is 2767. If you still want to see it more precise information, you can go towards a smaller scale. So you can see all this actual EEG. Actual EEG does not go beyond 500 or something. Scalp recorded EEG maximum 500 will for your blinks and all this thing. Otherwise, you have some movement artifact and all. Normal EEG, you can see this all can be considered as a normal EEG. You can hover over this and you will get a value here. This will give you a time value, this will give you amplitude, and this will give you which channel you are hovering. Like let's say you are hovering here on channel number 1 at 68.1504 second, you are getting 0.09 microvolt. So this again depends what you are checking, where you are checking, and all this particular thing. But you can see also event information is being recorded. Now we have a good enough of representation, so we can go ahead and do some processing. So now we need to tell them, okay, before event consider this time, after event consider this time. That is called epoching, very important. And you have to take minus. Why should we take minus time? That is because when you are working on any particular EEG or ERP extraction, you need to consider a baseline. When that event occurred, what happened prior to that. So for that we need to consider this particular minus time interval. For now I am just giving it 50, you can give it 100 also. It's called pre-stimulus baseline correction. Okay, but baseline correction is important. So that bean epoch, this is bean epoching stage, and this also there is a pop epoch bean function which does this particular thing. So yeah, you can run this particular thing, it will generate the bean epoch and it will save as a new set. So this third data set will be replaced by fourth data set. So you can see here this is your bean epoch. Okay, next step is, now again we will see how the data has changed, right? At every, you can see the earlier things have gone, this dashed line has come which corresponds to minus 50. And then further it has stayed till 400. The other in between data has gone. Okay, it has only saved the data which is like in the range, and it is same for all channels. You can see for bin 4 also it has epoched, for bin channel 4, channel 5, everything it has been equally placed and kept. Okay, I will there was a one instance of channel 3 as well. Yeah, this again I will show it to you. B1 is there, this pink one is B3 is there, right? And similarly B2 also we have seen before. So same ways all bins have been epoched as per the given time range. You can see it here, you can verify it throughout the recording, also bin 2, right? So now again let us go back and check. Some of the bins have values which is very high, which is not neural. So you can apply a simple voltage threshold and remove those bins. Let's say we will keep the amplitude range of minus 75 to 75. This is like based on your experiment, you can take any particular range, okay, and any particular as in you can take minus 50 to plus 50, minus 100 to plus 100, and for all three channels. So even in one of the channel it goes above or beyond this particular limit, it will discard that particular epoch. How many epochs out of all the epochs have been discarded, that you can see in MATLAB command window. How we can see, let us see quickly. So this is it will perform the artifact rejection, and again automatically saved with -AR prefix, which you can see here. So now you can you want to see, right, how many epochs have been rejected. So there were 3 bins. First bin 768 accepted out of 800. So 96% accepted. Second bin 100 out of all 100 accepted. Third bin 95 accepted. Now I have given plus or minus 75. The total rejected trials are 37 out of 1000 and 963 were accepted. If I give plus or minus 50, then there will be more rejected. right. So that is a trade off. You have to decide, there are so many parameters, what should be the filter frequency, what should be the epoch, bin epoch range, and all this thing you have to identify. After this thing your data will look even better. Let us quickly see the data once. So initially, as I mentioned, some burst and all, right. So you see those all are shown with this ochre yellow color or brown color. I will show for more time range so you can see the difference between them. See, all this thing have been vanished, where all this data or epochs have been accepted. Whenever it goes beyond some non-neural range, it will be discarded like this. Okay. So yeah, again one small illustration, it is happening, and artifact rejection has been performed. You can check it, there will be like 1000 epochs, and out of this 32 have been rejected. Last step is to generate a computed average ERPs. So that will be saved as a final, this you can select which data set you want to average here. Again what are your parameters and all, you have to exclude epochs marked during artifact rejection. So this is important, you should not include all the epochs, because you have identified that which particular epochs having non-neural high amplitude, so that you can remove, and again you run it. Once you run it, it will generate something called ERP, you can save it as S1. Okay. So it will stored a value in ERP. ERP is one structure which shows how many channels are there, how many bins are there, what is the data, what is the time range, etc. So now in ERP set there will be one ERP. Data set has all the operations which we have performed, S1, filt, EList, bin epoch, artifact rejection, and then here ARP. How it looks like, what are the waveforms. So we can quickly see that using plot ERP waveform. When you open the plot ERP waveform, there are several things. You want to plot all bins means standard target restrictor, all channels means there are 3 channels. You want to plot all 3 channels. Again you want to scale auto y axis, you want to show SEM. SEM is very important thing, once it really appears I will tell you, it is shows the variability, how many rows you want, how many columns you want, all this thing you can tweak it and make the things more, your format, you can change it, make your event more, make your results more publishable. Okay. So these are your final result. I mentioned this is about P300, right. So if you can see it, around 300 millisecond, this blue line and red line peaks, right. 300 means not exactly 300 it should peak, but somewhere in between 250 to 400, 200 to 400, that is a peak, and compared to this black line is very hardly visible here, but that is your standard. So whenever this rare event comes, your brain reacted to that and you are getting this kind of response. So this is the overall idea. You can see there is like here it is a time axis and it is in millisecond, this is your amplitude, and minus 50 to 400 it has been plot, and SEMs are also separable. SEM is standard error of mean, during 1000 of trials how much the variation occurred. So in between this and this, SEMs are also separable. Also when it comes to target, the response is much spread out compared to the response for distractor. Again you can play with this, you can identify what should have happened in between this example also. For that you can check your imaging, ERP imaging. So there are tools for that as well, you can check it. Also one more thing is you can have utilities, and you can generate your own data using utility. Simulate EEG ERP alpha version is there. So for different things, how many channels data you want, and if you do not have your own data, there are many students who have asked me that we do not have a provision to record EEG, how can we learn this thing. So first thing is there are online data sets available. Second thing is there are many conferences which gives you data and opportunity to do several signal processing algorithm on that, and you can trust the data set, like trustable data set which you can play with and learn so many things. Apart from that, you can simulate your data from here and get the things done. EEGLAB is a free software, you can install it with your MATLAB. Okay. So this is like a brief overview. Another point is, suppose you have some data and all, you can explore different commands in MATLAB. One more thing I wanted to tell you before I finish this lecture is you write EEG history. Whatever operation we have done, it will come as a command line interface. So this is for one file. Now by just one normal communication or batch file processing code, you can do the same operation for all the files. and you can run a code. So you can just run the code, go for lunch and come back. By the time all the recordings would be ready, all the results would be ready, which should be sent to a doctor or neurologist. So yeah, that whatever we have done here in terms of your processing, same thing can be done using code as well. So GUI and code mapping. For higher number of subjects, this batch file processing is important. Otherwise you can go ahead and do the same thing whatever I have done with using GUI. So this is a brief overview. If you want to use MATLAB for some other applications also, there are multiple apps based on your different problems. Considering this neural course, there are neural nets and all this clustering, all the classification, machine learning, and also corresponding on-ramps are also available. So you can just check it whenever you are free. Also there are plethora of signal processing applications are there which you can play with, not only for neural engineering, for overall biomedical engineering, and also for your normal solving problem. Here you can see image processing, right? There are so many problems of image processing and computer vision as well, which you can learn it and go into as much detail as you want. So again, it's a C. You can swim by yourself as much as you can. The objective of this particular small quick module to identify or to make you enable that how we can do all this thing. What is EEGLAB, what is ERPLAB? It is very important. If some small computation or something which will be asked in some assignment or question exam or something, okay, you should not be feel alienated or think, oh, what is this? So yeah, that is the overall idea. So I hope you have understood all this thing properly. If you have any doubts, you feel free to write to me in the forum, or you can reach out to me using this email ID. I will probably meet you in some other course or for some other module. Till then, if you have any question, as I mentioned, feel free to write to us, and all of you, please take care. Bye.